We're going to start it. Why don't we, but all this will be edited out. Why don't we test your slides?
Yes, let's do that. I guess you're now seeing my screen.
Yeah, why don't you test the share slide. Oh, beautiful, 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 beautiful.
Yeah, I have sound in one of those. I want to see whether you can also hear it. Not this one. Here. Can you hear the sound?
Of the video?
Yes.
No.
Okay, so maybe like this. And now?
What kind of sound should we be hearing?
Motor cycle engine sound.
No.
Oh, but how should I do then to reproduce the sounds as well? You know?
Yeah. I can't say I know.
Okay.
Say I know. Is the sound crucial?
I mean, in a way it is. Not
Okay.
entirely. I mean, I can just describe it, but it was
Yeah.
just nice to hear it. But yeah, I don't know then how. And if I just click on the link here directly.
Yeah, we might try to open a new, yeah.
And like this, do you hear it?
I do not hear it. Does anyone else hear it?
Like this?
No, I hear you really loud, but I do not hear it.
I don't hear it at all, sorry.
Yeah.
All right, then
Kimberly,
without sound.
I see you're on. Do you have any idea about playing a YouTube video within a slides? Kimberly's our president, so she might know these things.
You might have to try the sound setting at the top since you're the host.
Since I'm the host?
Whoever's the host
Yeah.
might have to look up on the sound settings
Okay. Let me look.
and then under the meetings.
Oh, okay. Let me see. Okay. I see meetings. I do not see anything about sound settings. Hmm.
So if you go under zoom.us at the very top of your screen and you go to meetings,
Yeah, I have zoom.us.
and then go to the the next thing is the meetings, and then there's a mute
Right.
audio.
Yeah. But mute audio then just muted me. All right. I see exactly what you're showing me, but all it says is mute audio. It doesn't say any other thing about audio.
Try it in, did you try presenter's view? For the video, for the PowerPoint?
You know, try that, Adriana.
Mm-hmm. But I guess here in Google Slides, I can only use it like this, I guess. So I don't have two screens where I have the presenter view. Let's say. No, it doesn't give me the option here.
Okay, I think we'll live without it and just say imagine.
Yes. I mean, it's
yeah.
just engine. People know how the engine sounds.
Like imagine this. Yeah, I think it's a choice between the speaker presenter voice versus the video presenter voice. Yeah. But yeah, I definitely like am in the top part of zoom.us meeting view edit window help, and I see nothing about sound.
Okay. I was also checking, couldn't find, but
yeah.
But otherwise, it works well, right? Everything.
Yeah, no, and your voice is very clear, so we can we can be happy about that.
Good.
Okay. All right. We'll let that go. All right. So I think we're good. A student named Alexis Chargo is going to introduce you.
Yeah.
So I will just turn it over to Alex, and then Alex will come on.
Yes. Yes.
Welcome, Parisa. So for everyone on the screen, why don't we stop sharing now? Like,
Yep.
Yeah, we're good. We're good. Okay. All right. Welcome. We are definitely not starting till 9:30. So if you want to take a little break, feel free. And we are recording this. So what time is it in Germany?
It is 3:22 now
Okay. Okay.
in the afternoon. Yeah.
Great. Okay, let's see. Hi, Alexis.
Good morning.
Good morning. You ready for your?
I am.
Okay, I just want to clarify your best last name. Should we
Mine?
go? Yeah. Chargo?
You hit, yeah,
Okay.
it's Chargo. No, it's Chargo. It just hasn't changed on here.
I was freaking out. I was like, do I not know her? Okay.
No. It's me. It just
All right.
the married name hasn't switched over.
Got it. Okay, well, that is helpful. Very helpful. And congratulations. So
Thank you.
this is Adriana. I don't know if you know her.
I have not met her.
So
Hi, Alexis.
Hi, it's nice to meet you.
Nice meeting you too.
yeah, so as it will go, I will welcome, then I'm going to turn it over to one of our trainees for the welcome, then Alexis will go, and then Adriana, and then everyone else
Sounds
should
good.
mute. It's fine. So we
Perfect.
sometimes these days on Zoom get a lot of people at our colloquia. So
Mhm.
we'll see how it goes today. It's our first one.
Ah,
You're our kickoff. Yeah.
Okay.
And
It's a huge responsibility then.
this is one of my students, Connie, coming on. from the Intro to Gerontology class I'm teaching. Hi, Connie, welcome. Hi, you're muted, but it's okay. Great to see you this morning. Also, just as an FYI, when more people come on, like, this is all being recorded, and then we will edit out these first kind of welcomes. Hi, Gail, welcome. Welcome, everyone. Welcome, everyone. We'll start in a few minutes.
Just wanted to say good morning to Adriana. Looking forward to your talk.
Hi, Anna. Thank you.
Hi. Yeah.
Yes, Anna, thank you for suggesting us to reach out. Welcome, everyone. We'll start in a few minutes. Okay, I think we'll go ahead and start. If that's okay. I want to welcome everyone to today's presentation. My name is Tam Perry, and I'm the new training director at the IOG. Welcome everyone to the second presentation of the IOG's Fall 2022 Colloquium and Professional Development Series. Alexis Chargo, one of our trainees, is pleased to introduce today's speaker. So I'm going to turn it over to her. And I do want to also say our next talk is on October 11th from Dr. Seoyoun Kim on the link between social engagement and health. But right now, we will turn it over to Alexis.
All right, good morning, everyone. Today I have the pleasure of introducing you all to Dr. Adriana Ruiz-Rizo. Dr. Ruiz-Rizo is an associate researcher at Yale University Hospital within the neurology department. Her research aims to understand how both structure and function of brain networks underlie cognitive and behavioral changes in pre-dementia. More specifically, her current research focuses on understanding functional brain connectivity, visual processing and attention in a healthy aging population, and subjective cognitive decline. Her research has been funded by the European Union's Marie Sklodowska-Curie Actions, and she is a recipient of several renowned international science and academic awards. Dr. Ruiz-Rizo's work has been published in prestigious journals, including Neurobiology of Aging, European Journal of Neurology, Neuroimage, Psychological Science, and European Journal of Neuroscience, among many others. In addition, she serves as a peer reviewer for a number of different journals. She was a recent outstanding postdoctoral researcher here at Wayne State's Institute of Gerontology and continues to collaborate closely with the Connect Lab here at Wayne State. Dr. Ruiz-Rizo is joining us this morning to discuss findings from a recent studies looking at visual processing in healthy aging adults. So if we could all please take a second and join me in welcoming her today.
Thank you, Alexis. Also, thank you, Dr. Perry and Dr. Doherty for this great invitation. And I'm going to go ahead and share my screen to start my presentation. So as Alexis already said, I will be talking today about visual processing speed in the aging brain. And I'm really delighted to share with you this research, which has accompanied me over the previous 4 or 5 years. So first of all, why am I interested in visual processing speed? Visual information is crucial for many activities of daily living. It is important for navigating the world, for acquiring new information, interacting with others, understanding the world, like when we see signs and symbols, performing actions or making decisions. And it is also not surprising because we are a visual species. If we see, almost all of our brain is devoted to visual processing. And that's why also it is not surprising that processes like aging or neurodegeneration that are also distributed processes can impair visual processing at some point. So I have focused mainly on 2 research questions. One is how can the aging human brain efficiently take up information from the environment? And the second is how can we use that knowledge to understand early indicators of cognitive decline? I'm going to focus on the first one first. We can assess visual processing speed with, for example, neuropsychological tests, like the one we see here, in which we have symbols and numbers. And the task would be to fill up these boxes with the symbols according to what is up here. And we can give an examinee 90 seconds, for example, to complete this task. And then we count the number of boxes that were completed. And this will be a measure of visual processing speed. However, The problem with this task is that it can involve other cognitive processes, like for example, memory. If you have better memory, maybe you can complete the task much faster, but it doesn't mean that you are necessarily have faster visual processing speed. Also, it depends on motor speed. If you're maybe have some slowness at the motor level because of any other reason, then you might be slow in this task, but it doesn't mean, again, that your visual processing speed is reduced. So overall, researchers have little control when using these tasks to measure visual processing speed. Now we can turn to computer-based tasks. For example, using these reaction times tasks, in which stimuli are presented in a computer screen, and we ask our participants to press a button every time they see the stimulus. Now, this sounds great, but again, it relies on motor speed. If you are slow because of a disease or a particular condition, then you will appear to have reduced visual processing speed, but in reality, that will be reduced motor speed. And also, we have little control about the perceptual threshold, or how much time somebody needs to actually be able to see a stimulus. So if we want to accurately measure visual processing speed, we need more precise measures or more precise tools. And to do this, we can turn to a theoretical framework to study visual processing speed. We can think of that as, for example, when we see an image like this one, we can imagine that objects are competing for becoming visually represented. This is a sort of competition in which they are all in a parallel race. However, this competition is not uniform, it's biased. And this is biased by attention. Attention would then be a selection mechanism. And depending on what we are attending to, then those objects are going to win the race. And so selection can be directed towards particular objects or to particular features. In this image here of the city center of the city of Munich, for example, if we're looking to take the suburban train, we will be looking for a particular sign, this S sign. Or if we wanted to find a restaurant or a particular store, then we will be attending different signs. In this case, this will be the category signs or these features. Visual processing speed in particular refers to the selection of features, so this aspect of attention. Now, in the lab, to more precisely study visual processing speed, we can use a visual report task in which arrays of letters are presented under different exposure durations. And because of the theoretical framework, we know that these letters will be competing to become visually represented. Where? In visual short-term memory. And so the task is for participants to see these letters, and we ask them to report all the letters they are fairly certain they have seen. And that's why this is known as a whole report task. How we quantify visual processing speed? We do it through a specific measure, which is the number of letters per second. This is a speed measure, very similar to the tachometer that tells us our speed in miles per hour. But in this case, our measure of speed will be in number of letters per second. I'm going to show you here an example of what the task looks like. So there is first the instruction for the participant. Now this will be the time when the participant reports the letters. Next trial. Again, the participant reports the letter. So you see, we can maybe see it again if it was too fast. So the idea is that we present these letters under different durations, and then we ask the participants to report all letters, but then we don't make any emphasis on speed or order of report. Again, this was slow. and this was faster. So we do this over and over again to obtain performance, different measures of performance, which we then use in the theory of visual attention model. And in this model, we try to model performance accuracy or report accuracy as a function of exposure duration, because it is expected that with increasing exposure duration, so the longer the stimuli are on the screen, then the mean accuracy will also be higher, and this is an increasing exponential curve. From this model, we can derive three main parameters. One is C, which is the parameter of interest. This is visual processing speed that reflects the rate of visual information uptake in a specific unit of time. This is the slope of the curve at the origin. And the origin is T0, which is the visual threshold for conscious perception. This is a measure in milliseconds that varies across individuals and indicates that time below which we cannot consciously perceive any stimulus. And finally, we also have K, which is the maximum number of elements that can be simultaneously represented in visual short-term memory. This is the asymptote of the curve and represents the maximum capacity of visual short-term memory. Using this paradigm, previous studies have tried to study and understand the relationship between aging and visual processing speed. And they have found this linear decrease in visual processing speed with increasing age, especially in older age. But also this has been shown even starting in the 20s. There is this more or less linear decrease in visual processing speed. And so that's why then the question becomes interesting to know what are the neural correlates of visual processing speed first, and whether these neural correlates can help us understand why there is this age-related decrease in visual processing speed. So this has been one of my main interests and one of my projects. So I have turned to study brain connectivity. So to explain what brain connectivity is, I would like to make an analogy of the brain for those of you who maybe are not very familiar with cognitive neuroscience. So let's think of the brain as a bunch of flowers. Now we can think of the flowers as the gray matter, or this darker part of the brain. And we can think of the white matter as the stems of these flowers. This will be the lighter part of the brain. So we can use brain connectivity focusing on either the gray matter, so different brain regions, for example, or we can study the white matter or structural connectivity. In more practical terms, in the real world, we study brain connectivity, or one of the methods that we can use to study brain connectivity is magnetic resonance imaging, or MRI. As I said earlier, we can focus on gray matter or white matter, and more technical terms are functional and structural connectivity. Functional connectivity, which will be focusing on the flowers, relates to the correlated activity between different brain regions, whereas structural connectivity that reflects or refers to the stems in the flower analogy reflects the white matter connecting those brain regions. In the next two studies, I will focus on functional connectivity. In this particular study to find brain correlates of visual processing speed in aging, we assessed 91 healthy participants between 20 and 77 years old. They were assessed with the whole report task, similar to the video I showed you earlier. And we focused on the cingulate opercular network as a potential brain correlate of visual processing speed in aging. Now, this name sounds a bit strange, so It just merely reflects the anatomical regions that are most important for this network. In particular, the anterior cingulate cortex. This is how the name takes the singular part. And then we have the opercular that refers to the frontal operculum or to the frontal opening that we see here, that lets us see the insula, which is this hidden lobe here in green. And this means that the insula and the anterior cingulate cortex are the core regions of the so-called singulo-opercular network. And this is, for example, all the regions that compose this network in this particular sample. We examined within this network which regions decreased with age in terms of connectivity. And one of those was the insula, one of the core regions. Now, when we look at the correlation between the connectivity in the insula and visual processing speed, we see a positive correlation, meaning that higher functional connectivity was associated with higher visual processing speed. And this was the case across all age groups. And furthermore, we found that decreased functional connectivity in the singulo-opercular network mediated the association between age and visual processing speed. So what we had expected from the beginning. These results imply that age is not deterministic, meaning that just a particular age does not determine the particular level of visual processing speed, so that there is room for improvement if we, for example, look at the level of functional connectivity. And the fact that just age alone does not determine the level of visual processing speed, that means that we can use tools or specific training to improve visual processing speed. In particular, alertness, based on the theoretical framework that we use, is a very important component for visual processing speed that we can use to actually improve visual processing speed in older age. And this is what the next study was about. We wanted to investigate whether alertness training could improve visual processing speed in older adults. Alertness refers to the brain's readiness state, so how quick we can respond to stimuli, or we can give responses. And this can actually be trained, for example, using computer tasks. This work was spearheaded by Dr. Melanie Penning. She did it during her PhD, and I collaborated with her in this project. I'm going to show you a short video about what the training task was about. So now there is a motorcycle sound that I guess you're not hearing. You have to brake when you hear the sound in order for you not to crash with any obstacle, like in this case, this falling tree. Again, you continue driving, you hear the sound, there is no obstacle, you don't need to brake. Continue driving, hear the sound, you have to brake, but you don't, so you're gonna crash. And this was the task, very simple, just people were sitting in a room and they were doing this game in several sessions. I'm gonna show you now the outline of the study. Oops. Yes, so we did this in 2 studies. The first one, there was a pretest session in which, again, we used the whole report task to obtain a baseline measure of visual processing speed. Then we randomly allocated older adults to different groups. They could be either part of the training group, which did the task that I just showed you with the motorcycle, or they could be part of the passive control group. This was just the group that didn't have any intervention, just did the pre-test and post-test. And the active control group, which was a group that had a different type of training. This was working memory training. And then after that, there was the post-test session in which, again, full report task was used and visual processing speed was obtained. Now, this training lasted between 4 and 6 weeks, and there were 16 sessions of 45 minutes each. What we expected to see was an increase in visual processing speed in the post-test after training, but specifically for the alertness training. And again, this goes back to the theoretical framework that poses the influence of alertness on visual processing speed. In a second study that involved additional 29 healthy older adults, we measured the baseline level of functional connectivity, and again, pre-test and post-test, but this time there was only alertness training. And we wanted to investigate whether the baseline level of functional connectivity in the singular perculate network was associated to a greater improvement or a greater increase in visual processing speed after alertness training. So these are the results. We can see here in red the alertness training group, the group of interest. We see in blue the passive control group, the group that didn't have any training, and in yellow, the active control group. This was the group that had the working memory task. Now, you see here that only the alertness training group had an improvement or an increase in visual processing speed at post-test, whereas neither the passive control group nor the active control showed any improvement. What does this tell us? First, the fact that the passive control group didn't improve means that it was not just, so the improvement we observed in the alertness training group was not due just to retest of visual processing speed. The fact that we didn't observe gain in active control group means that it was not just due to training, to any training, that we observed this gain in visual processing speed for the alertness training group. In other words, this increase in visual processing speed was actually specific to the alertness training group. Another way to present the results is through these box plots, where we can see the individuals here as these black dots, and we see them separately for each group. 0 in this case means no change after training, and we see that there is variability fairly across all groups. However, only the alertness training group showed this average towards overall gain, whereas none of the other groups showed that change, positive change. Now, in the second study, we could very gladly replicate the results of the first study. As you see, the trend here was very much like we observed up here for the red group, meaning again that in this second sample, we also observed an increase in visual processing speed after alertness training. And again, looking at the individual variability, but overall as a group, there was a positive change in visual processing speed. Now, turning to the brain data, this is the singular perculate network in this sample, all these yellow, red blobs. And we found one particular cluster located in the medial superior frontal gyrus that significantly correlated with individual gain in visual processing speed. More specifically, higher functional connectivity at baseline was associated with higher change or higher improvement in visual processing speed. And so then this again tells us that the variability we observe in the level of gain after this cognitive training can be explained by the level of functional connectivity. So to answer the first question, how can the aging human brain efficiently take up information from the environment? One answer based on this research points to the connectivity in the singular perculate network. because it explains the decrease in visual processing speed in aging, as I showed in the first study, and it also predicts the visual processing speed enhancement after alertness training, as I showed in the second study. Now, how can we use that knowledge to understand early indicators of cognitive decline? So this was a time then when I joined the Institute of Gerontology and the Connect Lab, led by Professor Jessica Damoiseau, and I investigated subjective cognitive decline. For those of you who don't know what subjective cognitive decline is, this is just a subjective perception of worsening in memory or cognitive abilities. This is then characterized by memory complaints. People are concerned about those complaints, those decreases. There could be depressive symptoms associated because of the loss that is perceived. People fear that they could develop dementia later on. But the puzzling thing is that there is normal cognitive performance. So even though they perceive that something is changing, that something is getting worse, still they do very well in cognitive tests. So then that's why I feel this is a very intriguing topic to investigate, because we have to find ways to objectivize these subjective perceptions. I've already shown you how important visual processing is in our daily activities. So that's why I think it is very important also to study how subjective cognitive decline affects the visual processing in general. And that's why one of the first studies at the Connect Lab was to investigate whether older adults with subjective cognitive decline process visual information differently in the brain, and whether they show more memory problems to complex visual stimuli. So for this, I analyzed a sample of 36 older adults, 16 of which had these memory complaints and concerns. They were assessed in the MRI scanner, in which they were shown pictures of scenes of the real world, indoor and outdoor, and they had to indicate whether those scenes were indoor or outdoor with a button press. This was the idea to encode the information for later recognition. Then, after this scanning session, there was the retrieval phase, in which they were shown the scenes they saw in the scanner previously, but also there were other new scenes intermixed, and they had to say whether the scene they were presented with was old or new, and whether they were sure about that response. Now, we made use of a very interesting property of images. So this is what we see. We just see photographs of real world places like this kitchen or this bedroom. All good so far. But what we don't see is that these images can also be categorized differently, for example, depending on the number of unique objects that compose those scenes. In that case, we can say that this image above would be a high complex scene, because it has a higher number of unique objects compared to the scene down here. So we made use of this complexity property of images to analyze whether this difference could have some influence on the way older adults store and recognize scenes from memory. So for the first question, whether older adults with subjective cognitive decline process visual information differently in the brain, we focused on two brain regions that are relevant for visual processing. These are the parahippocampal place area and the lateral occipital complex. The parahippocampal place area, you see in their name, it is relevant for processing places. And you can find it in the lower back part of the brain, whereas the lateral occipital complex is relevant for processing individual objects, like it gets more active when it sees individual objects. You can also find it in the ventral, very low posterior part of the brain. So we focus on these two regions, but we were expecting to see effects in the parahippocampal place area, because we thought if we were showing scenes, then we expect differences there. This is what we found. Here, I'm showing the brain activation, the level of brain activation for high and low loads scenes. We found that indeed there was more brain activation for high loads than low loads scenes in both the parahippocampal place area and the lateral occipital complex. However, we wanted to see that older adults with subjective cognitive decline actually process it differently, but this is not what we found. So the difference between older adults with and without subjective cognitive decline was not significant, meaning that they didn't seem to process visual information differently in the brain, at least in these particular brain areas. Regarding the second question about whether they show more memory problems to complex visual stimuli, this is what we found. This is recognition accuracy. This was in the retrieval phase. And we observed that overall high object load scenes were recognized better than low load scenes. However, again, we didn't observe any difference between older adults with versus those without SCD. However, it is different when you are shown, say, an old scene and then you have to recognize it versus when you're shown a new scene and then you have to make like further efforts. And this is reflected, for example, in the false alarm rate, which focuses on the new scenes, because you see a new scene and then you have sort of to encode it and compare it with the previous scenes you have stored. And for the false alarm rate, which is when somebody says that a scene is old, when in reality it's new, older adults without subjective cognitive decline showed lower false alarm rates, so better performance for high load scenes. In contrast, older adults with subjective cognitive decline did not show any difference, meaning that for them, it didn't matter whether the scenes were high loads or low loads. They didn't make that distinction, whereas the controls or older adults without subjective cognitive decline did benefit from the high load scenes. This told us that subjective cognitive decline seems to reduce the benefit given by perceptual detail in these high complex or high object load scenes. So this could imply that maybe SCD could somehow affect visual processing speed and or the cingulo-perculate network. We've already seen that there is this association between functional connectivity in the cingulo-perculate network and aging, but we don't know yet anything about the stems of the bunch of flowers. So we don't know much yet about the structural connectivity of the cingulo-perculate network. And it is important because the white matter could give some support to this connectivity in the gray matter or between brain regions. So that's why we thought that maybe it would be important to focus on the white matter in frontal tracts. For example, this one that is called the forceps minor. It could be important because it connects or it gives some substrate to frontal regions which are part of the cingulo-perculate network. We also don't know how the relationship between subjective cognitive decline or memory complaints and the cingulo-perculate network connectivity is. And we also don't know how the time frame, like what happens first, what happens next, is knowing that this is a dynamic process, both aging and cognitive or memory complaints. So in the next study, in the Connect lab, I tried to integrate all these components. And we did so in this model, this latent growth curve model. And this might seem a bit abstract here. So I'm gonna try to walk you through this. This was a study conducted in 69 older adults, also at the Connect lab. Functional connectivity was measured over 3 years, every 18 months. So that's why we have 3 measurements here. We also had measures of memory complaints or subjective cognitive decline. We had measures of structural connectivity or white matter connectivity. And we also looked at the interaction between memory complaints and structural connectivity here. And we also took into account age. Now, we wanted to see the effect of all these variables on 2 latent variables that are unobserved, but we know that exist, and that also can capture very well what happens across time points. And these were the mean functional connectivity and the rate of change in functional connectivity. We were interested specifically in the effects of memory complaints on the mean and the rate of change in functional connectivity, but also in the effect of the interaction between memory complaints and structural connectivity on the mean functional connectivity. This is what we found. So to make it clear here, the effect, so we found that the interaction between memory complaints and structural connectivity on the mean functional connectivity was significant. None of the direct effects from memory complaints on the mean or the rate of change were significant. Now, to make a bit clearer what that interaction meant, we found that the relationship between functional connectivity in the singular percolite network and memory complaints depended on the level of structural connectivity or the white matter connectivity. And more specifically, for those with high structural connectivity, or let's say preserved white matter, there was no association between functional connectivity and memory complaints. However, for those with lower structural connectivity, or let's say less preserved white matter, we found a negative correlation, meaning that greater memory complaints at baseline were associated with lower functional connectivity 3 years later. In other words, structural connectivity we found it was a moderator of the relationship between functional connectivity and memory complaints. So for the second question, how can we use that knowledge to understand early indicators of cognitive decline? One answer is that visual processing can help us understand memory complaints, because we have seen that subjective cognitive decline reduces the benefit of perceptual detail in scenes for memory recognition, as I showed in the first study. And second, SCD or subjective cognitive decline reduces the functional connectivity when structural connectivity is low. And I didn't show that directly in the model, but we found separately this correlation between structural connectivity and age. So in summary, what I've shown you today is that we can estimate an individual visual processing speed capacity, for example, using the theory of visual attention and the whole report task and modeling. Visual processing speed decreases with aging, but improves with alertness training. And also I've shown you that connectivity in the singular percolite network supports visual processing speed, both the decrease in aging and the improvement with alertness training. I've also shown you that subjective cognitive decline predicts connectivity reduction given a probably age-related decreased white matter. And finally, that subjective cognitive decline can interfere with the detailed visual processing, reducing the benefits of perceptual detail in scenes. And with this, I'd like to end my presentation. I'd like to thank all my mentors and collaborators. I'd also like to thank the funding agencies and you, of course, for your attention. And I'll be very happy to take any questions.
Thank you so much, Dr. Ruiz Rizzo. We really appreciate this thorough talk that really was presented in such a nice way to follow through the science. I think we can open it up for questions. If people want to either raise your hands or just let us know if you have a question. All right, I could start with one question. I guess as it would be nice to know sort of what the next projects or where this work can go, what you're thinking of. So if you wanted to share that with us, that would be great.
Yes, definitely. So one follow-up for this would be studying mild cognitive impairment. So, like the next, what we think subjective cognitive decline could lead to. One of these would be mild cognitive impairment, which is a higher risk stage for dementia. And we want to see how visual processing, but also other visual attention functions, because from this theory, as I showed you in the beginning, we can focus on objects or features. So we can use other visual attention parameters to also better understand visual processing. And so my next project will have to do with seeing the functional and structural connectivity correlates of another parameter, which is top-down control. And this reflects how the ability to focus on targets and ignore distractors, and to see how that makes it easier to remember information, like in the visual scene task. And this project actually is about to start, because I got it approved last week also, so it's going to happen, this particular project. And another one will be to study what I showed you with the high load and low load scenes again, but this time using a multivariate approach, for example, making use of machine learning techniques to decode brain representation when we're seeing these high load and low load scenes. So taking the same research aim, but through a different approach that maybe could be more sensitive, because we think there could be differences in visual processing at the brain level in older adults with subjective cognitive decline. But we think that maybe the approach we used for analyzing the brain data didn't allow us to see in a very sensitive manner those differences. So those are very crucial follow-up projects on these ones. Yeah.
Thank you so much. Other comments or questions?
I'm happy to jump in unless anybody. Fantastic presentation, Adriana. It really was a tour de force of so many different approaches that kind of all come together really beautifully. And I'll follow up just on the high load, low load scene, which I refer to as a high complexity, low complexity, which is really great that you kind of completed that project. I wasn't aware. And I think it's really interesting that you find the difference by subjective memory complaints. And I wonder how you think about it with regard to effect that we see in development. In children, we find that children find it most difficult or children's performance are specifically different. For the low complexity, they do well. The high load, they do not as well as adults. And then, so how would you put this together with what you find in aging, and in specifically the different with subjective memory complaints?
Thank you, Dr. Elfen. That's a really great question. And I was expecting the same. And this is the way for example, I posed the question when I said about the research question for that project. I was actually expecting that, what you observed in children. But I think it's related to this curve with visual processing speed, that first there is an increase and then decrease. So that's why sometimes, even though development and aging are, part of this continuum, still they reflect very different processes. So we would like to extrapolate what we see in children and development, and we would hope to see the same when there is, I don't know, some pathological process or pathological aging, but this doesn't seem the case. And I believe it kind of also matches this change. I don't know if you remember this curve here of visual processing speed that also goes in opposite directions. I sort of thought about it in that way as well. So like here that you see first this improvement, but then you see the opposite pattern at the other extreme of the continuum. So again, for children, it could be easier to recognize these low complexity images, whereas for older adults, it could be then the other way around. I guess, yeah, it would be those different processes.
Yeah. And I think it's really fascinating because it might be an opportunity to kind of put sort of dissociate at some level things that are more maybe working memory, because this is a number of unique object categories. You kind of maybe need to keep in mind more information versus aspects that are related to the perceptual processing of perceptual information.
Right.
And those are two different mechanisms that equally could be impacted by aging.
That's right. I first want to try the visual processing speed mechanism first. Although if I use this paradigm, still I can obtain a measure like the parameter K, which also reflects not working memory, but at least short-term memory capacity. So still we obtain both. We can see to which parameter it relates best.
Yeah, that's nice. Great work.
Thank you.
Are there comments or questions? Well, I think we'll just go ahead and thank Adriana for her wonderful presentation. I think it's great that you're from the IOG family, and we've come to see how your work is still continuing, and it's so important, and it's so connected to the work you did at Wayne. And I would like to thank everyone for joining the colloquium today. I understand there was a problem with the Zoom link, so apologies there. We will try to straighten that out. Join us next time, which is October 11th, for Dr. Seoyoun Kim's talk on social engagement and health. And I would just welcome everyone to join us this whole season of Colloquia series.
Thank you.